Okay, so let's take 2. Last one failed. Let's see if we can get this thing to open for me. No, doesn't want to play ball. Alright, we'll just go small screen. So, linked genes, so genes occurring on the same chromosome, that described as being linked. Looking at gene mapping, the idea of sex-linked inheritance, and then pedigrees. And I have actually, a couple of years ago, made a whole lot of little podcasts about each of the pedigree types. So I'll find those and upload those as well, so you can make use of those as often as you like. So, linked genes. Of course, if you've got a chromosome that's, a big chunk of DNA, there's going to be more than one gene loci, locus, position on the chromosome. So each chromosome has many genes, and they tend to get inherited together, of course, because the chromosome gets inherited in one piece. And so we talk about linkage between genes and gene trains, where a number of genes get inherited as a group, and we see these traits in medical conditions and things, or my facial expression, at present, well, not expression, but look, the beard, the shape, the eyes, eye colour, hair type. My sons have it, my father had it, photographs of Simpsons all over the world. We'd rather look like this. So there's clearly a group of genes that are determining how we look, that are just inherited as a group, with small variations, obviously, as people marry in. But of course, crossing overs also occurs. So when those two pieces of chromosomes sitting side by side, or within close range, get entangled, then we get that bit of recombination happening, which can break up those gene trains and change the linkage, the way things are inherited together. As you can see here, there's the chiasma. In fact, there's double link, double crossover there. And so I've actually chunked up those chromosomes, those genes, quite a bit. My head's in the way, so good. So I'll just get that out of the way a little bit. So we went from having X, Y, Z, big X, big Y, big Z being inherited together, and little x, little y, little z being inherited together. Those two chromosomes intertwine during meiosis, and we get a bit of crossing over happening. So now we have 4 quite different chromosome possibilities. Big X, little y, little z. Little x, what's that? Big Y, big Z. Little x, no, big X there, wasn't it? Yeah, little x. Yeah, you see what I'm talking about. So that recombination, of course, creates that opportunity for much more variation. And this crossing over allowed us to, for a long time, to map gene locations. Now, when your course was written, my emails are going to be coming in, isn't that wonderful? When this course was written in 2006, people had stopped using gene mapping in this form with crossover recombinations, because it had mapped the genome, and there's this, oh, well, we know where everything is, we've got all nucleotides. And then, of course, people thought to themselves, well, hang on, but where are the genes? Which gene does what? How far apart are they on each chromosome? And so gene mapping has once again become a technique used by geneticists. And so while it did disappear off the exam, it has sort of reappeared a little bit. So we'll need to talk about how that. I'll show you the notation for that in class, because I think I've got it in the slide. Go back there. So you can actually work out how far apart these loci are by the percentage of recombination between them. I'll do an example in class, because you need to see how it's done. You don't need to be able to do it on the exam, but quite often over the last few years, the notation for how you write it and the understanding of how it occurs has become a little bit important. So you know about it and don't get shocked about it. I'll explain it all in class. We also have genes that are found on the X and the Y chromosome. So that means they are sex linked in their inheritance. If you find something on a Y chromosome, then only males can have it. Only males have Y chromosomes. So if you're a male with a Y chromosome and this gene's on it, you're going to, your dad gave it to you because your mum can't. With the X chromosome, it's a little bit more complex because, of course, being male, we have one X from our mothers, and being female, you have 2 X chromosomes. So it gets a little bit more complex with the X chromosome. But again, because males only have one X chromosome, if you've got something like red-green colour blindness, which is a classic condition for this, males tend to show it more because it's recessive. You only need one allele to show it. So if it's a recessive trait and your mother passes it to you, then you'll pick it up. So mothers tend to be carriers. They have the condition, but may not express it because they're heterozygous, but they can pass it on more easily to their male children than to their female children, because if the father's not red-green colourblind, he can only pass on not colourblindness, the dominant trait. And we'll do a few little examples, do some Punnett squares, and I'll explain that a bit better. It's nice to have the screen, a whiteboard to write on and do stuff. For some reason, my Wacom tablet won't work with this laptop. Must try to get that fixed, actually. So, X-linked dominant, one of the sex-linked inheritance traits. Clearly, females only need one copy to show it. Males only need one copy to show it. Father will pass this trait to his daughters, which is a nice little clue. And heterozygous mother will have a 50-50 chance of passing it to all of her children, regardless of their sex. There's little things you need to know when you're working out the exam pedigree. I'll do some pedigrees soon. Sex-linked on the Y, clearly males pass it to males. So most of the chromosomes on the Y, most of the genes on the Y chromosome are about male fertility and sexual characteristics, like growing beard and things. Deep voices. Well, that's all testosterone linked, so that's probably wrong. Anyway, pedigrees. Now, I pinched this off the web years ago. I should probably try and find a better one because it's got a mistake in it. And it's American. We don't quite use the same notation, but essentially you'll see this notation on your exam paper. So male, square, females, circles, put a line between them, and they're making babies. And then here's the babies. We can have what we call dizygotic and monozygotic twins. So dizygotic twins are two eggs met with two different sperm. So they're non-identical twins. They're dizygotic. And so you show them in this way. If you get a monozygotic pair of twins, it's one egg, one sperm, have made a baby which at blastocyst stage has separated. So the 16 cell bodies become 2 8-cell bodies and then developed. And of course these are identical twins. So you can see a small problem here. These are the monozygotic twins, but they're boy and girl. That would suggest that they aren't exactly identical. So there's a mistake here. You can find sex unspecified. Clearly in most human ones that doesn't occur, especially these days. But in the past, lots of abortions and miscarriages and things would occur. Stillborn children, and you may not have recorded the sex because the child was never baptized and christened and recorded as a birth. And so you might have a birth, but a sex unspecified. More commonly found in animal pedigrees where you're not too fussed about particular traits. Clearly in really big pedigrees, which you would have for animals, rather than worrying about listing every single animal, you might just say 4 boys, 3 girls for that particular coupling. The questions you get in the exam won't be like that because you need to be able to work things through. They'll always look more like this. Affected females are coloured in, affected individuals are coloured in, so this is either affected male, affected female. Carriers, we don't tend to use this notation, and you certainly won't get it on your exam because, of course, they want you to work out who the carriers are. This American system seems to suggest that for sex linkage, we use a dot in the middle, and for anything that's autosomal, we colour in half. The ones I've seen in the English tradition seem to only use a dot. Death, you put a line through, obviously that makes sense. Stillborn, the little shrunken one. Proband, so the arrow points to the individual that is being described. Contiguous marriage, you've got a brother and sister here making babies. Not good. Don't do that. And we shouldn't do it with animals either, of course, or worry about all that inbreeding stuff. But that, of course, does happen in the breeding of animals, by error or deliberately in some cases. Not that's a good thing. What you're going to see is this sort of notation. You're going to have first generation, one big I for first generation, II for second generation, so using Roman numerals, and then numbering 1, 2 from left to right. So when you're describing what's happening in a exam answer, you need to talk about the individual big I number 2 was a female carrying the condition. Therefore, she is homozygous recessive or whatever. But it's a way of describing who you're talking about, which means your examiner knows who you're talking about and knows you understand what you're doing. So you'll be careful with the language in that one. And then we have some classic formations. So autosomal recessive. So here we have 2 generations skipped, but we have a mother in the first generation, and then her great-grandchildren suddenly show this condition she has, red hair or something, green eyes, some strange condition that doesn't get seen too often. It's a recessive type. And so you need 2 copies to show the condition. And this means in here we have lots of carriers coming through. So we would say a carrier there for sure, carrier here for sure because they're direct descendants of that person. And then we imagine that this male here also carries that condition. Let's say green eyes. And so 2 of the children end up with green eyes, although neither parent show it. So fairly equal distribution between sexes and a big gap. Really indicative of autosomal recessive. Autosomal dominant, you tend then to find the condition in every line because you only need 1 copy. So you're going to find lots of people with it, fewer without. Again, you're looking for reasonably equal numbers of male-female, so it's not showing any real difference there. And then you just describe it as autosomal dominant. Really good exam trick to play with you is the lethal gene, where in this case big A dies without being born. So the homozygous dominant doesn't exist. So your genotype ratio is now 0 to 2 to 1, and your phenotype ratio is 2 to 1, and you're expecting 3 to 1. So it's just a little bit of data that throws you out. That's a good little trick to try and muck this up. X-linked recessive. So in this case, we're seeing a really clear demonstration of a grandfather giving this to this grandson through his daughter. So the daughter's got big X, X big A, and dad's given the daughter little a, and that little a has been passed on to this son from this daughter. So the female's a carrier. We're seeing 1 sex, and we're seeing few individuals. So both things are saying 1 sex, X-linked, few individuals, recessive. The hardest one to find when you're doing these, and actually I've made some up as crap, it's a really bad example, I must make another one up. The hardest one to determine between X-linked, dominant, and just dominant is this. So the biggest thing to look for is a predominance of 1 particular sex. Now when I was making this, I loaded the data too hard. There should be some females in here to make this a little bit more achievable. But essentially you're looking at 2/3 to 1/3 male-female. And again, we're finding it in every line of the pedigree because you only need 1 copy to show it. Now I've made some other podcasts to work through some of those, and I'll find those and put them up. This is all about variation. We're going to talk in Area State 2 about the need for variation for natural selection to work. This is all about producing variation. We talk about variation in 2 forms, a discrete variation and a continuous variation. So eye colour is essentially discrete variation, or height's even better. No, height's not very good at all. Male-female, discrete. You're either one or the other. This is really good for height. We've got you to line up across the back of the classroom. We have a nice little graph showing this range from shortest to tallest. And so there are multiple alleles and multiple genes involved in achieving this, and so we get this continuous variation. There might be 1 gene loci like XY producing this outcome. These 2 diagrams come off exam papers. So this is the sort of question you might get. Discrete, 1 gene loci. May only have 2 alleles or 3 alleles. Continuous, many alleles, many genes, and greater variation as an outcome. And variation is important. I'm going to run out of time, aren't I? This last slide, so that's right. So how do we get variation? Lots of alleles, lots of genes, so polygenes, polyallelic, really useful. Crossing over, of course, during meiosis creates lots of differences. The environment, so the influence of the environment changes things. The independent nature of assorting for sperm and egg